are now going to return to our gut feelings, and our next speaker is Johanna Lampe, who I've actually known for, I don't know, 10 years, more than that, give or take, a decade. And she is a wonderful researcher. So she is a professor of nutritional science and epidemiology at the UW, and also a full member in the Public Health Sciences Division at the Fred Hutch. She studies the mechanism by which components of the diet, we're talking vegetables here, kids, particularly constituents of plant food, which is vegetables, alter your susceptibility to and risk of cancer. So can broccoli save you from cancer? She knows the answer to that. Her primary research activities include controlled dietary interventions in humans to determine how individual variation in biochemical response to high plant food, read vegetables, may explain differences in disease risk. Can you tell I didn't really like vegetables growing up? All right, well, I'll get over that. The Lampe Lab also studies the effect of diet on gut microbial community and the relationship of the gut microbiome to biomarkers of cancer risk. Now, many are asking, what is she doing there? She is surveying the vineyards of Saint-Cern on the Loire River in France. And I guarantee you there was a bottle of wine involved in that. So Johanna earned her Ph.D. in nutrition sciences with a minor in biochemistry. So right off the bat, she's really smart. She trained as a postdoc fellow in epidemiology in Minnesota, and then earned her MS and RD in nutrition and dietetics from the University of Minnesota. And even tonight, she said, I'm so glad I don't live in Minnesota. She did. It's like -20 there. It's horrible. She serves on grant review panels for the NCI, National Cancer Institute, and American Institute of Cancer Research, and she's a senior editor for a journal that's called Cancer Epidemiology, Biomarkers and Prevention. So that goes on to say how smart she is. So she's going to talk to us about diet and the human gut microbiome, and whose diet is it anyway? Johanna.
Good evening, and Terry, thank you very much for that kind introduction. It really is a pleasure to be a part of mini med school here this evening. I think being a nutritionist by training, I recognize the importance of nutrition in your medical school training, and given that this is mini med school, we will cover it in 40 minutes, and which some physicians would say was about the extent of their nutritional training too, and the rest of their med school. But so let's get started. So when we think about diet, and in this case when we talk about diet, we're really talking about the totality of food intake and everything that's part of your nutritional uptake, not necessarily diet as in a weight reduction diet per se. It could be a very different types of dietary patterns, different food choices, etc. But we recognize that what you choose to eat can play an important role in disease risk, particularly for many chronic diseases, such as cardiovascular disease, diabetes, as well as a number of different cancers. And in this regard, we tend to think about this, if we're going to try to divide this up, a couple of different ways. Certainly the amount we eat, i.e. the amount of fuel that we take in that helps to maintain our energy, helps to keep enough meat on our bones, and sometimes a little more than just meat as we add extra layers of fat and in relation to energy imbalance, this really has become a problem in the Western countries. And we've recognized that particularly with regards to disease risk, obesity and adiposity are an important part of the dietary story. In addition, we also have a whole host of compounds, which we just here are calling dietary constituents. That is all those various chemicals that are important for our health, both from the standpoint of vitamins and minerals, many other bioactive compounds that aren't necessarily essential for life, such as nutrients, but can play a role with regards to health. And what we're recognizing is that this whole gamish of components of diet are heavily influenced by the gut bacteria, both from the standpoint in affecting our fuel availability, as Dr. Damon pointed out, that gut bacteria may play a role in adiposity, as well as their effect on handling a number of components of diet. Similarly, the gut microbes can have direct effects with regards to disease, and we've heard a little bit about that this evening already. And the flip side of this is our diet also plays a very important role in determining the composition and the activity of the gut microbes or the gut bacteria. So sort of a two-way street here, and we need to consider both sides of the story. So what I'm going to do in the next 40 minutes then is to talk about several aspects of this diet-gut microbiome relationship, and to focus mostly on what are the gut microbes doing with our diet, how does this affect what we are actually exposed to as human beings. We put something in our mouth and we think that this is going to provide us with certain nutrients, and we're all set to go. Well, maybe we'll have a few discussions about how that may not be the case. And then ultimately, how does this influence our health and our risk of disease? So when we think about microbes and disease, you've already had a number of presentations talking about microbes as infectious agents. And coming from a cancer center where we focus on their potential role in cancer, we know that they may account for about 20% of cancers worldwide with regards to cervical cancer, liver, gastric cancers. And these, in some cases, are very much direct effects on the cells in these different target tissues. However, as you've heard, microbes can also have a number of other effects as modifiers of physiology. That is, they can affect how body cells behave. And in the context of our diet story, also we're going to focus primarily tonight on microbes as modifiers of exposure. That is, they metabolize a number of dietary constituents, they metabolize drugs, they metabolize many of the compounds that we think of as being carcinogenic, and they also affect energetics and obesity. So this will be the piece we focus on. One thing that we're always challenged with when we try to understand the relationship of diet to disease, and then also its interaction with the gut microbiome, is that human diet is extremely complex. Unless you're living on one food morning, evening, and lunch, you are going to be exposed to thousands of different compounds at each meal. And also how you prepare your food, how you chop it up, and what size pieces do you put it into. All of these things can affect the availability of the nutrients as well as the other compounds within food. And this really becomes an issue when we start to think about the impact of the gut microbiome on this, because if you're consuming things in a form that structurally is not available to the enzymes in your intestinal tract to break them down, many of these then become food for the bacteria. So just thinking about whether or not you consume wheat as a whole grain versus you grind it up versus you process it further, all of these can affect how it's handled by gut microbes. So there's no question about it, we are what we eat, and I think this picture here does a nice job of showing this. This was work of Ruth Lays from Cornell University now, and she took poop samples from just about every animal she could come across, either in zoos or on the African plains, and examined their gut microbial communities. And when you rely on the computer approach to clustering these, what you find is that all of the herbivores are tending to cluster together, that is, those who are only eating plant materials. The carnivores down here, both the dogs as well as the large cats, are clustering here, and the omnivores, who eat a little bit of everything, are over here. So really, what the gut microbial community is very much driven by what's available in order to feed it. And I think one of the real fascinating things about the gut microbial community is its metabolic capacity. I like to think that it's designed in some regards to make the most of what could be a really bad situation depending on what your diet is. In that when we think about food going in, we have a whole host of enzymes and processes that we can use in our own intestinal system to be able to digest our food. And what's really left then for the bacteria further down the intestinal tract are what we might call the indigestibles or the leftovers, or things that we didn't chew well enough to break apart cell walls in plant foods. And so the bacteria have really are relying on a whole host of reactions that we don't have the capacity to carry out, but that allows them to handle many of these what we would consider undigestible components. Probably the most familiar is fermentation of carbohydrate that allows them to obtain energy out of their food products, a variety of other reactions such as reduction reactions, esterification, aromatic fission, which means breaking ring structures, and a number of other hydrolysis reactions. So these are all things that they can do that we can't, and that's sort of part of where those extra thousands of genes that they've got that we don't have gives them the one-up on us as far as being able to handle all these components of our diet. And when you look at the different genes that are involved in the gut microbiome and where the gut microbes put their action, a lot of it is really in this area of handling many bits of our diets in relation to carbohydrate metabolism, that is a lot of the fermentation reactions, amino acid metabolism, again related to protein degradation, and then a whole host of reactions that are involved in what we call xenobiotic degradation, which includes many of the plant compounds that we often think of as being potentially beneficial, as well as pyrolysis products, which are what you get if you let your meat flame up on the grill when you're preparing dinner. And as we've talked about earlier, also a number of drugs that can be handled by gut microbes. So this is really, they're putting a lot of energy into activities to handle these various substrates. Probably the one area that we've spent most time focusing on in the past has really been the fermentation. And this relates to how we handle dietary fiber that humans do not have the capacity to digest themselves, and it's really the microbes that are doing all the work, breaking down these very long chains of sugar molecules that then release shorter ones and ultimately produce what we call short-chain fatty acids that have a whole range of bioactivities, and some of which I'll talk about in a few minutes. But these include acetate, propionate, butyrate, and then the famous gases that come when you've eaten a very high-fiber diet, and include carbon dioxide, hydrogen, and for the 30% of you out there who are methane producers are also producing methane. So these are the products of fiber fermentation. There's a lot of interest in the short-chain fatty acid butyrate because it has a wide range of activities. We've known for many years that it serves as an energy source for colon cells, which is very important. You want to keep the cells involved in maintaining the lining of your colon healthy and happy, and so this is really an important part of it. But we also know it has a number of other signaling effects as well. It can affect our satiety, so it will make you feel fuller if you are producing more butyrate. Certainly plays an important role with regards to reduction in inflammation in the gut and reducing oxidative stress. We know from a lot of animal studies that butyrate is important in decreasing tumor production, and it also is important in maintaining the gut barrier function to prevent things from slipping through between cells and causing more systemic problems. So this is actually a short list. There are a growing number of studies showing the importance of butyrate for a whole host of pathways related to health and disease risk. Microbes also are very good at handling a number of protein degradation products. So if we think about proteins as being chains of amino acids, the amino acids are the building blocks of protein. the microbes are able to break apart the proteins into the various different types of amino acids. For those of you who don't chew your steak well enough and just sort of swallow it whole, a lot of your bacteria are going to see a higher amount of protein as a result of you not being able to sufficiently digest it, and it'll hit the colon and contribute to microbial metabolism. So we have what we call aromatic amino acids. These are amino acids that have ring structure that form an interesting group of phenols and indoles. Sulfur amino acids can produce a number of sulfur compounds. And then we have all the other amino acids that undergo, again, a number of bacterial metabolic processes to produce some useful things and some not so useful things like ammonia, a number of amines, organic acids. So one of the interesting things that comes out of metabolism of the aromatic amino acid tryptophan is the production of indole. And as I said, the aromatic amino acids have these ring structures, and the bacteria have a particular enzyme that allows for production of indole. Interestingly, we're finding that indole plays a major signaling role with regards to affecting the production of either pro or anti-inflammatory signals. Again, it can also, like butyrate, strengthen epithelial cell barriers and also decreases pathogen colonization. So here's a compound that's being produced by our gut microbes that is actually helping to benefit the host with regards to the health of the gut epithelium. On the flip side of this, when we look at metabolism of the sulfur amino acids, we get generation of hydrogen sulfide. That's stinky, egg-smelling gas. And this is produced by the gut bacteria through fermentation of amino acids such as methionine, cysteine, cystine, and taurine, and results in the production of hydrogen sulfide, which has been shown to be toxic to colon cells, both in vitro as well as in vivo and intact animals. And there's some data to suggest that it may contribute to inflammation and risk of ulcerative colitis and colon cancer. Just to give you an idea of what happens if you're consuming large amounts of protein containing sulfur-containing amino acids, this was a study that was conducted coming from nutrition. We do studies where we feed people things and see what happens. And in this case, they took 5 individuals and gave them 5 different diets of different amounts of protein that ranged from 0 to 600 grams of meat per day. In case you're wondering how much 600 grams of meat is, it's over a pound of meat a day. So probably not the usual intake of most individuals here, but what they did then was to collect all the stools for the time that people were in the study and looked at the amount of sulfide present in the stool samples. And you can see as you eat more meat, you produce more fecal sulfide, suggesting, again, from the standpoint of producing a rather noxious compound, not a good thing. There are a number of other enzymes. Again, these are specifically bacterial enzymes that can produce noxious compounds, such as conversion of nitrate from preserved meat products to nitrite, which can glom onto a whole host of other compounds in the body, particularly in relation to complexing with DNA, can form adducts, which are not appropriate forms of the DNA and cause DNA damage and contribute to cancer risk. So not a good thing, which is partly why we often, in the context of colorectal cancer, say avoiding large amounts of meat and processed meat may be important, partly for this reason. Another story related to bacterial handling of dietary constituents is in relation to cardiovascular disease. There are a number of food products, particularly, again, animal products containing fat, that are good sources of what's called phosphatidylcholine. And when you consume these, the choline is converted by the gut microbes into a compound called trimethylamine, which in and of itself is not a terrible thing, but when it's then absorbed, taken up in the liver then adds an oxygen moiety to form what's called trimethylamine N oxide, or TMAO, which has been shown to be associated with atherosclerosis and a number of cardiovascular disease-related illnesses, as well as death. So what they found in trying to study this and the impact of the gut microbiome on this is that we know that when you give people more phosphatidylcholine, you see more production of TMAO, and if you give people antibiotics, you get a decrease in TMAO production, partly through preventing the conversion of choline to trimethylamine. So if you take, and then when you take away the antibiotics, the production of trimethylamine N oxide goes up again, suggesting that in this case, the bacteria are very important in that first step of, in the production of TMAO. When we look at individuals' levels of plasma TMAO in relation to cardiovascular disease, this is looking at 3 different groups of individuals. This was among a study of over 4,000 people undergoing a cardiac catheterization procedure, relatively healthy at the time when they were being evaluated, but then followed up for 3 years later. And what we see is that those individuals have the highest, who are in the group with the highest TMAO levels at day 1, 3 years later are most likely to suffer from either a myocardial infarction, stroke, or death due to a cardiovascular event. Again, showing this is not a good relationship, and anything that we can do to help to reduce exposure to this compound may be important. Enough sort of negative things about the gut microbes. On the more positive side, thinking about their handling of many of the other compounds that we're exposed to in our diet, unless of course you're Dr. Brentanol and don't eat your vegetables, or your fruits for that matter, we find that there are, again, even in the plant foods alone, thousands of compounds that can serve as substrates for gut microbes. And we'll focus today on a couple of these, but just to give you a feel for some of the examples, one group are the glucosinolate compounds in cruciferous vegetables. That is your broccoli family veggies. Yes, you should eat your broccoli. Isoflavones, or the phytoestrogens found in soy. For those of you who are beer drinkers and like your beer very hoppy, isoxanthohumol is a compound found in hops that is also metabolized by gut microbes into a very estrogenic compound. And also a group of compounds called lignans that are present in whole grains and vegetables that have been shown to be associated with lower risk of several cancers. Now, the interesting thing about these is that the activity of having consumed these foods is really dependent on the gut microbes handling them and how they handle them. And so we'll focus on the glucosinolates in cruciferous vegetables and the isoflavones as an example. So we like to think, or we pretty much have been hit over the head with all the data that have come out, that really the cruciferous vegetables are the poster vegetables for cancer prevention. Of all of the different groups of veggies, these are the ones that consistently show up as being associated with lower risk of a number of cancers: lung, colorectal, breast, prostate, and pancreatic cancer. So these are your broccoli, your cauliflower, your cabbage, radishes, etc. And we also know from experimental data that there are certain compounds that are present in the cruciferous vegetables that are contributing to these chemopreventive effects. The isothiocyanates and the indoles are the 2 groups of compounds, and they've been shown to prevent tumor formation, also been shown mechanistically to decrease inflammation and oxidative stress, and have a number of effects that are also relevant for reduced carcinogenesis. The challenge with this compound is that as we eat our vegetables, the important isothiocyanate that's present in our vegetables, and why your mother said eat your broccoli, is not the form that it's in the vegetable itself. The compound in the vegetable is called a glucosinolate, and it has, because it has a glucose molecule attached to it. And in this form, it's not active at all. What it requires is an enzymatic reaction that allows for removal of this glucose to produce the free isothiocyanate. Now, any of you who have consumed a cruciferous vegetable recently and consumed it raw may have noticed as you're chewing on your broccoli stalk or your radish that you'll get a sort of burst of sharpness or sort of a mustardy type response. This is really the action of the myrosinase enzyme in the broccoli or the other cruciferous vegetable, allowing for release of the isothiocyanate, which has this lovely sulfur moiety on it that contributes to that sort of effervescence in your mouth. And unfortunately, most of the time when we eat cruciferous vegetables, we cook them. So we really deactivate this compound that's present in the broccoli itself, such that we're not able to convert the glucosinolate to the isothiocyanate. And fortunately for us, our gut bacteria can handle this reaction too. So in many cases, we can rely on those gut microbes to take care of this reaction. So just by way of example, if you're consuming your broccoli sprouts as a source of isothiocyanates, this shows what happens when you consume broccoli sprouts uncooked, so i.e. the myrosinase is present and active. If you chew your broccoli sprouts very well, you can obtain more of the isothiocyanates from the broccoli sprouts. If you swallow your broccoli sprouts whole, you do not achieve the same benefit. Not to mention, you can imagine how much fun that is. But to make matters worse, when you cook your broccoli sprouts, i.e. you denature the myrosinase, you're again getting even less out of your broccoli sprouts in the way of the isothiocyanate. Whereas if you pre-treat with a source of myrosinase, that you can actually almost obtain the total amount consumed as can be recovered, and we can recover it in urine knowing that it's gone through the body and it's been available to the rest of your body's cells. So this really shows that depending on how you handle your foods, depending on how well you chew them, all of these factors can affect the availability of a compound that could be potentially beneficial in relation to cancer risk. And we see this in a large study that was conducted in China that was actually interested in whether or not a broccoli sprout tea could be used to help prevent effects of aflatoxin, which can bind to DNA and cause damage and contribute to risk of liver cancer. They showed that the more of the isothiocyanate that is being absorbed by a person contributes to less, is associated with less aflatoxin DNA addicts. And when you look at how much people are actually recovering, it could be anywhere from 1 to 45%. And this was with everybody receiving the same dose of the broccoli sprout tea. Now, similarly, I had a graduate student who was not afraid of poop and was very interested in this question of why is it that some people can extract a lot of the isothiocyanate from their diet and others can't. And he was interested into what role the gut microbes are playing in this. So this is just a histogram of the range of recovery of isothiocyanates when you give everybody the same dose of cooked broccoli. And it ranges anywhere from 1% to about 30% can be recovered, with most people being less than about 10%. So they're not getting a lot out of their cooked broccoli. So what he did then was to ask these people if they would kindly provide him with a stool sample, which he then incubated with the glucosinolate and looked at what happened when individuals who we identified as being good at excreting isothiocyanates, those are the folks in yellow, versus those that were not. And what you see, and this is taking stool samples, incubating them in a flask with media and some glucosinolate, and looking at the degradation of the glucosinolate. And what he found was that those individuals who typically put out a lot of the isothiocyanate in their urine, i.e. their bacteria seem to be pretty good at this, are also the ones that when you take their bacteria and put them to work in a flask, similarly are the ones that are producing larger amounts of the isothiocyanate. So important from the standpoint of the contribution of the microbes to this availability of this compound. Another story that has received quite a bit of attention is how the gut microbes process the isoflavones in soy foods. There are several isoflavones that are present in soy foods that have received the classification of phytoestrogens. That is, they have weak estrogenic properties. And like many other things, these compounds are heavily metabolized by the gut microbes. And one of these is dietsane, which can go through several reactions and produces two products, odesmethylengolinsen and equal. We won't test you on odesmethylengolinsen, which we call ODMA for short, just because nobody can pronounce it. But when we give people a source of dietsane, i.e. feed them with large amounts of tofu or soy milk, and look at their amount of these compounds that they're producing and excreting in their urine, we find that most people are able, or their bacteria are able to produce ODMA. So 80 to 90% of individuals, you find ODMA in their urine, whereas a much lower percentage of people excrete the compound equal. And this really varies by populations. Western populations, we usually find a prevalence of only 20 to 30% of individuals have the capacity to produce equal, or their gut microbes have the capacity, whereas in Asian populations, it's more in the area of 50 to 60%. So there are differences in the gut microbial community that allows for production of this compound. Why do we care? Both from the standpoint, and there have been over the last 20 years a lot of studies conducted trying to understand the potential benefits or possible harms of soy food consumption and the effects of soy isoflavones. And what we've seen in many of these intervention studies is that individuals who have the capacity to produce equal often differ from their equal non-producing counterparts with regards to handling of a whole lot of hormone metabolism, also downstream effects related to estrogen and androgen hormones with regards to the menstrual cycle length, bone mineral density, different gene expression in cells as a result of being an equal producer or not, suggesting again that the gut microbial community is playing some role in modulating the effects of these isoflavone compounds. Similarly, in observational studies and epidemiologic studies, again, we see differences in relation to the equal producing capacity. So there are a lot of factors in relation to the gut microbes that are coming to play. Interestingly, when we try to go looking for any one bacteria that's responsible for this reaction, we can't find it consistently across individuals, which suggests again that it's really a combination or a consortium of bacteria that's responsible for this overall reaction. And some people probably have a different set of bacteria that handle it as opposed to another individual and their particular bacteria. So a number of different species can handle these reactions. Looking at the obesity and fuel imbalance side of the relationship between diet and the gut microbiome, really exciting area and a lot yet to be learned from the standpoint of the contribution of the gut microbes. As Dr. Damon pointed out, there have been a number of studies in animal models showing that germ-free, in this case indicated by GF, as compared to those rodents that receive are either born with bacteria and provided bacteria at birth versus those that may have been germ-free or notobiotic until inoculated with a bacteria. The presence of bacteria results in a higher percent body fat compared to the germ-free. And when we look at specific areas of fat depots, again, the same thing that we see, about 40% more total body fat in the presence of the microbes. And there have been a number of studies that followed up on this, showing that in some cases the types of bacteria can play a role as far as increasing body fat. And this work is ongoing and is a very complex relationship between amounts and types and their effect. Another possible use for fecal transplants, this is a study out of the Netherlands showing what happens when you take stool from lean donors and provide them to individuals who have metabolic syndrome. That is, they're at risk for diabetes as a result of a number of metabolic processes, both with regards to glucose handling, et cetera. So this just gives you an idea of what happens when you start with a group of lean controls. This is looking at insulin sensitivity, and the higher that these bars are, the better off you are. So lean individuals tend to be insulin sensitive, in good shape. Individuals who are prone to metabolic syndrome before a fecal transplant are lower in both cases here. And in this study, they provided the individuals with the metabolic syndrome either with a fecal transplant from a lean person here, showing that this transplant resulted in improved insulin sensitivity, whereas if you retransplanted the same person with their own bacteria, no difference as far as response. So again, not only can you cure C. diff, but the importance of the gut microbes with regards to signaling in relation to insulin sensitivity appears to also be an important piece of this. Also, the investigators noted that you see about a 2.5-fold increase in the bacteria in the stool that are important for butyrate production. So again, butyrate pops up as an important signaling molecule in this context. So you really sort of think, how can the gut microbes be contributing to obesity? Definitely in the context of increasing availability of calories from your food, particularly on a high carbohydrate diet, high fiber diet, where you may not be able to digest a lot of the fiber, they are providing extra energy from that. But they're also having a number of effects with regards to how our bodies process the calories that we are consuming. There are some data to show that we have an increased stimulation of dietary fat absorption in the small intestine. So our microbes really want to help keep us healthy and alive, since that's where they're living. But from the standpoint of signaling in the small intestine, that this can be a piece. The short-chain fatty acids serve as energy. And then there's also another effect with regards to systemic inflammation that may be important for obesity, and that is the presence of lipopolysaccharide, which is an endotoxin from the wall of gram-negative bacteria, which can cause low-grade inflammation in the body, and this also tends to increase risk of adiposity. So this is an area that really is under intense study at the moment, trying to understand this, and there's going to be a lot more that we're going to learn in the next couple of years. So just to recap some of the pieces from this evening, gut microbes metabolize a variety of different dietary constituents. There are very few things that they don't seem to be able to handle, so they're always looking for a good source of energy in whatever form they can find it. From our standpoint, as the consumers of the food, our diet as we consume it is not necessarily what we're actually exposed to from the standpoint of the gut microbial metabolism. There are certain gut microbial communities that may predispose somebody to greater body fat gain, and we haven't quite nailed down which ones these are and how we can affect this, but certainly this is going to be an area that's going to receive a lot of attention. And finally, the gut microbiota can also mediate both host metabolism and obesity through a number of different mechanisms, and there's still quite a bit of hand waving going on here, but stay tuned because I expect there will be a lot more coming out also. So on that note, thank you for your attention.
Over there. I'm over here. Hello.
Oh,
Hello.
see, behind the camera, I can't.
Hey, that was great. That was very information dense. That was a great bunch of stuff. Hey, my simple question, we were talking about hand washing, et cetera, and splashing lots of chlorine all over hospital rooms. I guess my question, real simple question, is do some of these bacteria develop, immunity, sensitivity to chlorine? If they're, drug-resistant bacteria,
Yeah.
are there chlorine-resistant bacteria that we're going to be seeing in the next few years?
So far, C. difficile is not chlorine resistant, so that's the good news there.
Yeah, there's different mechanisms by which various agents will kill bacteria. So antibiotics, they can be very targeted and, against certain proteins that will mutate, and find a way around it. Whereas things like chlorine and alcohol-based hand washes are very non-specific mechanisms. So the likelihood of organisms evolving a way around those types of ways of cleaning are probably less likely.
So my question is regarding how you're saying how cooking vegetables denatured mayonnaise, which made it less good for us. Is that a common thing throughout all vegetables, or is there any real benefit to cooking our food besides meats?
There are a number of good reasons to cook your vegetables too, and it really is dependent on what kinds of compounds you're wanting to get out of them. For example, the carotenoids in green leafies and carrots, et cetera, they actually are more bioavailable if you cook them and if you cook them in oil. It actually improves the uptake and the absorption of those. So it's based on, and the bacteria can handle carotenoids too, but for the most part, we can absorb those more rapidly in the upper part of the small intestine. So it really is dependent on the compound and how it's absorbed into the body.
That was a great question because I don't like raw vegetables, so I love broccoli, but uncooked broccoli just does not do it for me.
And the other thing that you can do in relation to the broccoli thing is you can cook your broccoli, but also if you have a little mustard with it, or if you have a raw radish with it, you actually will be providing myrosinase at the same time that you're consuming your cooked broccoli, so that you are setting up that chemical reaction in your gut, as well as relying on your gut microbes, of course.
I want Johanna to cook my food. She's in charge of my diet from now
We have
on.
a cookbook. Can we start a cookbook this way? Go ahead, sorry.
And then would you say, suppose I didn't care whether it was cooked or non-cooked, is there in general better to do this than the other? Would you say better to cook it or not? I mean, you said it's kind of depending on the compounds, but in general.
I think partly one of the important things with regards to consumption of fruits and vegetables, and this may sound kind of very general, but here in the U.S., we actually don't consume as many of them as we should. And so to the extent that we can find ways to make them palatable and help ourselves to eat more of them, we're going to derive a number of benefits. And it doesn't matter if you cook them or eat them raw with regards to the possible benefits of dietary fiber. So it really, it's sort of specific as to particular compounds. But overall, we've developed approaches for handling many of our foodstuffs that have come up over centuries of food processing and handling, that many of those are there for a reason, as far as decreasing pathogen exposure, increasing bioavailability. So traditional approaches sometimes are the best ones to go back to.
All right, thank you.
Mm-hmm.
So thank you. Interesting information. The fecal transplantation I'm curious about. It sounds like really interesting research out there, but will it need to be repeated? Because with the diabetic patients and the insulin sensitivity, I'm wondering if you do the transplant, how long it will take before it gets corrupted again, since probiotics can help make our flora better. What about, returning to a previous diet, changing the flora again? Do you know what I mean? The cycling effect.
So the data's kind of young for use of fecal transplantation for management of like diabetes or obesity. That data is really young. I mean, we're just saying it's coming out this year. The data that's been probably among the longest out there would be the data looking at treatment for C. difficile. And so in that case, once you make the transplant, 95% of the time you're done. You don't have to make another transplant. It's really rare that you might have to go back and make another transplant.
Okay, very good. Thank you.
Do microbes play a role in gluten intolerance?
You're a GI doctor.
Not related to
It's an area
celiac?
of active research right now. People are looking at the microbiota and celiac disease to see if there's a dysbiosis there. But as you mentioned, glucose intolerance is a disease that's separate from celiac disease. It's actually a well-documented disease where there are studies showing that people eat bran muffins that have gluten in them versus the identical bran muffin without gluten. Those with gluten develop symptoms even though they don't have any mucosal inflammation. I am not aware of any data, but it does seem plausible that one mechanism that it could work through is the microbiota.
Describe the efficacy of the fecal transplant for certain problems. How common is it? Is it really here? If I go to my doctor with pains and he thinks that might be an answer, is it available?
You mean a stool transplant?
Yes.
Well, that's my next job is I'm gonna sell skinny stool. It's not here yet. If you have C. diff, it's here for you. If you have C. diff that you relapse. But in general, in the US right now, this is all experimental work, but it's really intriguing. And so you gotta stay tuned for the next mini medical school next year.
Thank you.
Is it known, you talked about the difference in digestion of soy in Asian populations versus here on average, and is that a matter of having more soy, therefore developing more? I mean, is it known whether the frequency of consuming soy impacts that, or is it other factors, or is that known?
It's not entirely known, and it seems to be a mix of different things. I think one of the things that we always have to remember is that in Asian countries, children, when they are weaned, are starting on soy, and so it really is a part of the diet early on in life. Often in the Western countries, we don't start in on it until we're a lot older. And that may be part of it because that's also the time that you're starting to set up your adult microbial community. And so that exposure at that point may contribute in part. Also, the types of soy foods that are traditionally consumed in Asia are different than many of our soy foods that are on the market here. And so the combination of the isoflavones and the form that they're in food processed soy food products here are different than in a traditional product in Asia, which can include some fermented products. So the whole host of different factors that may come into play, but it's definitely an intriguing difference that we see.
So I have a question about C. difficile. Does it infect people that are perfectly healthy, that have really good gut flora, or does it tend to get people that are already a bit sort of compromised in terms of the variety and balance that they have in their good and bad bacteria?
So some people just have C. diff in their gut, but it's in harmony with the other bacteria, so it doesn't grow out. And then what would tip you over is if your gut microbiome gets out of harmony, and that allows it to grow out. So it can be in there just as a normal little friendly bacteria, not causing trouble. It's when you get too much of it. So some people acquire it, and other people already have it.
So is it uncommon to have it as a normal bacteria, or is that fairly common? Is there a certain percentage of the population that has that?
Yeah, it is. It's reasonably common to see C. diff in the stool, or I don't want to say it's rare. Let me put it that way. It's not rare to have that in there, but the C. diff that gets you in trouble is you get a lot of it, and it makes these two toxins that poison you. And that's where you get into trouble. And so we know that things are amiss when you start making the toxins. So for us, if we were going to look at your stool and say, oh, she's got diarrhea, it's not enough for me to find the C. diff in there. I'm looking for the toxins. That's how I know you got trouble.
And like realistically, how long do those spores actually live? I have sort of a funny story about
They
that.
last months.
A month, okay.
Yeah, I'm interested in any of your thoughts on fasting and juicing.
Probably, it's not an area of my expertise. However, from a standpoint of the effects on metabolic syndrome and effects on insulin sensitivity, there are some interesting studies showing that intermittent fasting can actually improve insulin sensitivity. And what it's doing as far as your gut microbial community is concerned is a different story. There are some great studies showing what happens to a boa constrictor when he's fed versus fasted, and you can imagine boa constrictors fast for weeks on end before they get their next pig. But you see incredible shifts in the gut microbial community as a result of that. So you could imagine similarly with extended fasts that we will see changes in the gut microbial composition as a result of no more fiber in your diet, no more food, and your gut microbes are relying on the mucus that's being secreted by the colonic cells. And so they're really relying on other sources of substrate, and therefore different bacteria are going to grow and others are not. So there are going to be shifts, but what the impact of that is long-term on health, I don't know.
What about the juicing?
From a gut microbial-centric view of the universe, I would say, removing all that dietary fiber is really unfortunate for a microbe because fiber fermentation is an important contribution to support of the gut microbial community. When we're encouraged to be consuming 25 to 35 grams of dietary fiber a day, the juicing component and throwing away the fiber is sort of a loss in that regard.
Is the appendix seen as a repository storage facility for the microbiome? So if for some reason you do kind of get cleaned out, you've got a little storage in the appendix?
That's an interesting question. So there are very interesting associations with the appendix. So for example, folks that have had an appendectomy are less likely to develop ulcerative colitis. Also on colonoscopies, when you diagnose ulcerative colitis, you often will see, so ulcerative colitis is only in the left side of the colon, or the most distal part. Well, it can be the whole colon, but it tends to sort of migrate from the left and up and around. And interestingly, you can have a little area around the appendix that gets inflamed as well that's not contiguous with the rest of the colon. So there are interesting things happening there. Whether it's a repository for bacteria for repopulating the guts, or if it's more of a lymphoid function, it's hard to say, but there are some interesting associations with disease there.
I have two questions. The first is for the first is for Johanna about the graph you had on protein and the sulfur byproduct. Was that for just like a big grouping of protein, or animal specific and versus red meat or whatnot?
So the study where they fed the different doses of meat, is that the one you're talking about?
Yeah. So
right, so it was feeding meat, and partly because focusing on the sulfur-containing amino acids that are contributing to hydrogen sulfide production. So from that standpoint, different foods have different amounts of sulfur-containing amino acids, and it often tends to be the animal-based products that are going to be higher in the sulfur amino acids as compared to many of the plant foods that are sources of protein.
My second question is related to the juicing. So trendy wheatgrass enemas, do they do anything?
Sometimes I just cannot believe what people put up their butts. That is such a waste of good wheatgrass juice. And don't put your coffee up there either. That's a bad plan. No, skip that.
Okay. Well, I was going to ask, when you're doing an enema, does the stuff get up far enough to actually do any good? Because if you're doing a fecal transplant, is it beneficial only because it's getting up so far?
So it depends on the volume of the enema, how far up it gets, and then how much you roll around and put your legs up in the air. But in general, most enemas don't make it very far out of the rectum. And so that would be why most enemas are more or less kind of silly. And so, but you know, I've heard people, they have the high colonic, you've heard of the high colonic, and they take like a gallon of coffee and in we go, and you know, really in your heart of hearts, that's a bad plan. And so in general, high colonics are not going to help you. And little short enemas are also not going to help you unless you have a specific disease in your rectum that requires an enema for treatment. Like people have rectal problems where we put an enema in to treat that. But in general, you should drink your wheatgrass juice and your coffee.
Thank you.
Okay, one last question.
I'm just wondering more about the probiotics. They were mentioned as a treatment after you've had an antibiotic. But could you talk more about whether it would be helpful to use a probiotic daily or weekly, or is yogurt sufficient to renew bacteria in your gut, or what's the thought on all of that, just as a preventative health practice?
So the literature on probiotics is a difficult literature because, as you can imagine, there are many different probiotics out there. And so I'd mentioned those 4 probiotics, which have been studied a little bit more, and there are others as well which have been studied. But your typical probiotic that you get off the shelf, you know, from Whole Foods, hasn't been studied. And maybe the components within it have, but until very recently, FDA hasn't even regulated what's present in the probiotics. So you can't even really be certain that, A, that bacteria is there, and B, that it's active. Because a lot of, you know, the bacteria potentially could require refrigeration, or, you know, they lose their activity over time. So it's a difficult question to answer because there isn't a whole lot of evidence. But that said, for specific formulations, for specific diseases, there is good evidence. And because it's a relatively innocuous treatment, we'll often say, give it a try and see if it helps.
Well, thank you.
Thank you.